Okay, dear students, welcome to the today's class leprosy. So let us discuss about leprosy is also called as a Hansen's disease caused by Mycobacterium leprae, and this Hansen's microorganism is discovered by Hansen in 1874. This leprosy is a chronic non-fatal infectious disease. It affects mainly the cooler parts of the body such as the skin, mouth, respiratory tract, eyes, peripheral nerves, superficial lymph nodes and testes. And the main involved organs are skin and nerves. Other organs are liver, spleen, bone marrow and regional lymph nodes. Causative organism caused by Mycobacterium leprae, which closely resembles Mycobacterium tuberculosis but is less acid fast. The organisms in tissues appear as compact rounded masses globi. Mycobacterium leprae can be demonstrated in tissue sections, in split skin smears by splitting the skin scrapings from cut edges of dermis, and in nasal smears by following techniques. Acid fast staining technique, it is also called as a Ziehl-Neelsen staining technique. In this one, discolored by lower concentration 5% of sulfuric acid. So in less concentration of sulfuric acid, the microorganisms lose their color. That is called less acid fast, 5% sulfuric acid solution. Second test is Fate-Farragut staining procedure, is a modification of ZN procedure. So second test. Third test is Gomery methenamine silver GMS staining can be employed. Third test, GMS staining. Fourth one is molecular basis methods, that is polymerase chain reaction can be used to identify the microorganism. Fifth one is IgM antibodies to PGL1 antigen seen in 95% cases of lepromatous leprosy, 60% cases of tuberculoid leprosy. So here two types of leprosies are there, lepromatous leprosy and tuberculoid leprosy. Both can be identified by IgM antibodies to PGL1 antigen. Next, we'll discuss the slit smears technique gives a reasonable quantitative measure of Mycobacterium leprae when stained in ZN method. ZN method, we are going to identify the microorganisms and examined under 100x oil objective for determining the density of bacteria in the lesion. So that number of bacteria in a lesion is identified by the 100x oil objective. That bacteria is called as a bacterial index BI. Bacterial index BI is scored from 1 to 6, range from 1 to 10 bacilli per 100 fields. So for 100 fields 1 to 10 bacilli will be there. It will be that leprosy will be positive. If it is more than 1000, multibacillary leprosy. So multibacillary leprosy means microorganisms amount will be more. BI bacterial index will be more for the multibacillary leprosy, while bacterial index BI of 0 plus is termed paucibacillary, PAUCI paucibacillary. Then incidence, that means where it is reported more, leprosy is reported. This disease is endemic in areas with hot and moist climates and in poor tropical countries. Leprosy is almost exclusively a disease of a few developing countries in Asia, Africa and Latin America. According to WHO, 8 countries are where it is more endemic, that is India, China, Nepal, Madagascar, Nigeria, Brazil, Indonesia, Myanmar, also called as Burma. In these countries, 8 countries, 80% leprosy cases are reported. In India, the disease is seen more commonly in states Tamil Nadu, Bihar, Puducherry, Andhra Pradesh, Odisha, West Bengal and Assam. And next, subheading mode of transmission, how this leprosy is transferred from one person to other person. Leprosy is a slow communicable disease. Incubation period between first exposure and appearance of signs of disease varies from 2 to 20 years. Average about 3 years. So average incubation period for the microorganisms is 3 years. The infectivity may be from the following sources. First source is direct contact with untreated leprosy patients who shed numerous bacilli from damaged skin, nasal secretions, mucous membrane of mouth and their hair follicles. Second one is materiofetal transmission across the placenta from mother to the baby through the placenta. Next, third mode of transmission is from the milk of leprosy affected mother to infant. So transmission from milk of mother, leprosy affected mother to infant. Then immunology of leprosy. So what are the immunological molecules involved in the leprosy? In tuberculosis, the immune response in leprosy is also T cell mediated delayed hypersensitivity type 4 reaction. So type 4 immunological reaction will be taking place. In this immunological reaction steps, first one is antigens of leprosy bacilli. Lepra bacilli has several antigens. The bacterial cell wall contains large amount of Mycobacterium leprae specific phenolic glycolipid. Phenolic glycolipid PGL. Phenolic glycolipid means PGL1. Another specific antigen and lipoarabinomenin LAM. Lipoarabinomenin. Unique feature of leprosy bacilli is invasion of peripheral nerves, which is due to binding of trisaccharide of Mycobacterium leprae to basal lamina of Schwann cell. So due to binding of the trisaccharide of Mycobacterium leprae to basal lamina of Schwann cell. Next, second point is genotype of the host, genetic composition. Genetic composition of the host has known by MHC class HLA type determines which antigen of leprosy bacilli shall interact with host immune cells. HLA MHC class. Then third point is T cell response. There is variation in T cell response to the main forms of leprosy. Unlike tubercle bacilli, there is not only activation of CD4 plus T cells but also of CD8 plus T cells. So that means CD4 plus T cells are the major molecules playing role in the tuberculosis, whereas CD8 plus T cells are mainly responsible in the leprosy. CD4 plus T cells in lepra bacilli infected persons act not only as helper and promoter cells but also assume the role of cytotoxicity. So CD4 plus T cells are responsible for the cytotoxicity in leprosy. Two subpopulations of CD4 plus T cells that are also called as T helper cells. CD4 plus T cells are also called as the T helper cells. Th1 cells, Th2 cells. Elaborate different types of cytokines in response to stimuli from lepra bacilli and macrophages. In tuberculoid leprosy, CD4 plus T cells response will be more. Lepromatous leprosy, CD8 plus T cells response will be more. So overall conclusion is T cell response, CD4 plus T cells response will be more in the tuberculoid leprosy, whereas lepromatous leprosy, CD8 plus T cells response will be more. Next, humoral response, that is blood response. High levels of immunoglobulins will be reported, that is IgG, IgA, IgM, and antibodies to mycobacterial antigens. But these antibodies do not have any specific protective role against lepra bacilli. So do not have any protective role against lepra bacilli. That means these antibodies are not protecting the human body from the infective microorganism. Then for identification of microorganisms, they are doing lepromin test. It is not a diagnostic test used for classifying leprosy on the basis of immune response. That means this lepromin test is used to identify the leprosy based on immune response. That means in order to identify the class of leprosy. Intradermal injection of lepromin, an antigen extract of Mycobacterium leprae, reveals delayed hypersensitivity reaction in patients of tuberculoid leprosy. First point, an early positive reaction appearing as an indurated area in 24 to 48 hours is called Fernandez reaction. Second point, a delayed granulomatous lesion appearing after 3 to 4 weeks is called Mitsuda reaction. That means here reaction, what early reaction, that means 1 to 2 days, 24 to 48 hours is called Fernandez reaction, and later delayed granulomatous lesions 3 to 4 weeks is called Mitsuda reaction. So two reactions, Fernandez reaction, early reaction, delayed reaction, Mitsuda reaction will be observed after injecting the lepromin, an antigen extract of Mycobacterium leprae. Patients of lepromatous leprosy are negative by the lepromin test. The test indicates that cell-mediated immunity is greatly suppressed in lepromatous leprosy. So this test shows that cell-mediated immunity is suppressed in the lepromatous leprosy, whereas not suppressed in the tuberculoid leprosy. Patients of tuberculoid leprosy show good immune response. So immune response will be good in the tuberculoid leprosy, whereas immune response will be less in the lepromatous leprosy. Then next, classification of leprosy. Classification of leprosy by Radley and Jopling's classification. two main forms of leprosy are reported: lepromatous type, tuberculoid type. Lepromatous type low resistance, tuberculoid type high resistance. Again, these two groups, these are called as the two opposite poles of the immune response, also called as a polar forms of leprosy. So polar forms of leprosy means lepromatous type and tuberculoid type. And again, this is again converted into five groups based on their nature, that is borderline indeterminate type. So based on the borderline it is there, or indeterminate type is there, so based on that they are classifying as a 5 groups: TT, tuberculoid high resistance polar leprosy, then BT, borderline tuberculoid leprosy, BB, mild borderline leprosy, BL, borderline lepromatous leprosy, LL, lepromatous polar leprosy. So LL is low resistance lepromatous polar, whereas high resistance tuberculoid polar. So these are north pole, south pole. So we can, if you consider north pole, high pole, tuberculoid leprosy is having high body resistance against the microorganisms, whereas low body resistance in case of lepromatous leprosy. Difference between lepromatous and tuberculoid leprosy. So here features: lepromatous leprosy, tuberculoid leprosy. So features, if you consider: skin lesions, nerve involvement, histopathology, bacteriology, immunity, lepromin test. So first, skin lesions. Skin lesions difference will be there between lepromatous type as well as tuberculoid type. Lepromatous type, symmetrical, multiple, hypopigmented, erythematous, macropapillar, nodular. These are all leonine phases. So leonine phases of skin lesions will be observed in the lepromatous leprosy, whereas tuberculoid leprosy, asymmetrical, single or few lesions, hypopigmented and erythematous macular lesions will be observed. Whereas nerve involvement present in both lepromatous as well as tuberculoid leprosy, nerve involvement is there, but sensory disturbance is less severe in case of lepromatous, whereas sensory disturbance will be more in case of tuberculoid leprosy. Then histopathology: collection of foam macrophages, lepra cells in dermis separated from the epidermis by clear zone. So here clear zone will be there between the dead cells and the living cells. Lepromatous leprosy, hard tubercle similar to granulomatous lesion eroding the basal layer of epidermis, no clear zone. So no clear zone in case of tuberculoid leprosy, whereas clear zone in case of lepromatous leprosy. In bacteriology, lepra cells highly positive for lepra bacilli seen as globi, cigarettes in pack appearance multibacillary type. Lepra cells few seen in destroyed nerves as granular or bedded forms, bacillary type. So difference is there in case of bacteriology also. Here multibacillary type is lepromatous leprosy, bacillary type is tuberculoid leprosy. Then immunity is suppressed in case of lepromatous leprosy, so very less body resistance, whereas tuberculoid leprosy immunity will be more, so high resistance against the microorganisms will be there in the body. Lepromin test is negative in case of lepromatous leprosy, and lepromin test is positive in case of tuberculoid leprosy. The next variants. So types not included in Radley-Jopling's classification is indeterminate leprosy, pure neural leprosy, histoid leprosy. So these are the 3 variants which are not reported in the RJ classification, Radley-Jopling classification. Indeterminate leprosy, an indistinct non-specific stage of any type of leprosy. Here pure neural leprosy absent in the skin lesions, whereas present in the nerve involvement main. Histoid leprosy, so variant of LL, skin lesions resemble nodules of dermatofibro lesion are positive for lepra bacilli. Then reactional leprosy, based on shift in the immune states in patients of leprosy and treatment, 2 types are distinguished, type 1 and type 2. These 2 types in the reactional leprosy. Type 1 is reversal reactions, whereas type 2 is erythema nodosum leprosum, ENL. Type 1 reversal reactions. Polar forms of leprosy do not undergo any change in clinical and histopathological picture. Borderline groups of unstable, undergoing upgrading or downgrading of patient's immune state. Accordingly, there are 2 types of borderline reactions: upgrading reaction or downgrading reaction. So first one, upgrading reaction, characterized by increased cell-mediated immunity, occurs in patients of borderline lepromatous type on treatment who upgrade or shift towards tuberculoid type. So from the lepromatous to the tubercular type, shifting is called upgrading of the leprosy. That is changes in the leprosy. Then downgrading reaction characterized by lowering of the cellular immunity. Borderline tuberculoid type who downgrade or shift towards the lepromatous type. So from the tuberculoid to the lepromatous type, shifting of the leprosy is called downgrading, whereas from the tuberculoid to the shifting to the leprosy. So type that means if you see downgrading means tuberculoid shifting towards the lepromatous, downgrading, whereas in case of upgrading it is lepromatous shifting to the tuberculoid. Then type 2 erythema nodosum leprosum ENL characterized by tender cutaneous nodules, fever, iridocyclitis, synovitis and lymph node involvement. So this is about the two types of reactional leprosy: type 1 reversal reactions, type 2 erythema nodosum leprosum. Thank you.